I think we have a common theme in this symposium, and that is we talk about large and complex networks on all sorts of different scales. And for the next few minutes, I want to take you down to a very small scale, about 1/25th of a millimeter or smaller, and that's the world of cells, which also contain highly complex networks. All life on Earth is made out of these cells. A human being needs about 13 trillion of those. They can also work on their own. They can make all these other structures. And what's amazing is that a cell is nothing else than just a bag of molecules that interact with each other, and they interact with each other in highly complex ways. This is the protein interaction network of a quite simple cell. You see the cell in the middle here. This is about 5,000 proteins, often interacting with many others. And this is static, but of course the whole thing is very dynamic. And whenever we try to study signaling pathways in the cell, we actually find that it's frustratingly complicated. It's highly complex. So in general, in engineering, there's this principle that complexity is the enemy of reliability. Yet cells actually have both aspects. On the one hand, they are very complex in their regulation, and on the other hand, they execute this really reliably. And we are interested in this: how is reliability and robustness achieved in cellular function? And to study this, we look at a process that is essential for our life, that is highly dynamic, that is precisely timed, and that indeed has a complex regulation. And on top of that, it's beautiful to look at, and that's cell division. And what you just saw here is what normally goes on for about an hour, which was a mammalian cell dividing. We study this not in these mammalian cells, but in a simpler cell type, yeast, which, however, serves as a very good model for many other cell types. And we do that because we are interested in the basic regulatory principles. And that is much easier to understand in a simple cell type than in a metazone cell type, which has lots of added-on regulation that adds to the complexity and makes it even more difficult to understand. The imaging, as you see on the right side, is not quite as nice as it is in the mammalian cells, but we can image with a resolution between seconds and hours all the processes that we are interested in. When we study a process, we use whatever technique seems useful to us in order to address the problem, and we usually initiate collaborations to do that. So in the past, we have, for example, used chemical biology tools. We have used quantitative proteomics and phosphoproteomics. We have used even more advanced imaging than we can do in my own group. But one technique that we come back to again and again is computational modeling, because it's really helpful to understand how cellular signaling is wired. So we typically combine our quantitative experiments with a computational model for the process we are interested in, and then derive predictions from the model and test those predictions again in experiments. And we like it if the model works. We actually also really like it if the model doesn't work, because that tells us that we have not understood something fundamental and there is actually something to be discovered. So within cell division, we mostly focus on how do cells get the right number of chromosomes, which obviously is very important for the survival because they have all the genetic information that the daughter cells need. So we look at a signaling pathway that can detect if there's any problem with the chromosome attachment to the mitotic spindle, which would prevent them from being properly segregated. And we look at the process itself, how the chromosomes split and how they become distributed. And that also very closely links to the work of another VBI biology fellow who is not here today, unfortunately, Daniela Cimini's group, because they are interested in what happens to the cells once the chromosomes have missegregated and the cell ends up with a wrong chromosome number. So over the past years, with respect to the signaling pathway, my group has identified new links in this pathway. And as I told you, we are interested in how can it function reliably. And we have found that this pathway can only function reliably if protein levels are pretty constant. They do not fluctuate too much, and we cannot modify them much, otherwise the system breaks down. In this signaling pathway, we have also found other threats to its reliable function, which were surprising, but which are probably coming from a trade-off, because the pathway needs to be reliable, but on the other hand, it needs to be dynamic, and you cannot do everything at once. With respect to getting the chromosomes into the two daughter cells, that's completely different. So in that case, this step in the cell division is completely robust to large changes in the protein levels. And it was really difficult initially to understand why that is. And we have now started to figure out some of the mechanisms that allow cells to cope with such high variability and nevertheless really reliably deliver the chromosomes. So I've told you that we are interested in this aspect of robustness. How is cellular function reliably achieved? The flip side of that is the fragility. So in which aspect are cells or cellular function sensitive? And that links to two other areas. One is synthetic biology, because that tries to engineer new function inside cells, which often can be done, but often turns out not to be very reliable and robust. So very often does not work in different cell types or does not work in different environments. And we hope that what we find can inform these synthetic biology approaches. On the other hand, finding out where cell division is sensitive to perturbations can be very informative for cancer treatment, because as Carla already told you, cancer is basically just a disease of uncontrolled cell proliferation. So I would really like to thank my group, all our collaborators, all the other VBI biology fellows here at the Institute, in particular Daniela Cimini's group, with whom we have joined lab meetings. And our interests also really are closely aligned with the scientific interests of John Tyson at the Department of Biological Sciences, and Jean Pecou and Neil Adams here at VBI, with whom we also have joint meetings. And I would like to thank all my colleagues from the third floor and all the VBI staff who really make it fantastic to work at this institute. Thank you, and thank you for your attention.